Good afternoon, everybody. Welcome to the Society for Developmental Biology Postdoctoral Seminar Series. We are very excited to highlight the work of our outstanding postdoctoral members. So today, Cara Moravec from University of Wisconsin-Madison and Brandon Carpenter from Emory University will share their research. So each speaker will give a 20 minutes talk followed by 10 minutes of QA. So please enter your questions in the Zoom QA box. So I'm very happy to introduce our first speaker, Cara Moravec. Cara got her PhD from Stony Brook University, where she studied behaviors by using zebrafish as a model system in the laboratory of Dr. Howard Ziroctin. And for her postdoc, she joined the laboratory of Dr. Francisco Pellegrio, and there she studied the germplasm aggregation and compaction in the zebrafish embryo. And Cara has got a lot of awards and honors. For example, the most recent Excellence in Mentoring Awards. So today she's going to talk about the chromosomal passenger complex in germplasm aggregation during early development. So Cara, I'll let you start.
Thank you for that lovely introduction. And I would also like to thank SDB for giving me the opportunity to present our research on the role of the chromosomal passenger complex in germ aggregation in the early embryo. So one of the most important cell fates that is necessary for the survival of any species is the establishment of the primordial germ cell in the early embryo. In vertebrates, the formation of the primordial germ cell can occur by two different mechanisms. The first mechanism is inductive, where the germplasm generates signal that induces the formations of the PGCs, and this is observed in species including mammals. The second mechanism is by the inheritance of maternal products, specifically maternally derived germplasm, and this is observed in species including zebrafish and xenophores. In zebrafish, germplasm is inherited from the mother. It becomes aggregated into the furrow. These aggregates are necessary for the formation of PGCs, which then create germ cells, and then the cycle continues. So in the early embryo, germplasm is composed of both maternally expressed RNAs and proteins. These germplasm RNAs include RNAs that are necessary for PGC formation across animals, such as Vasa, Nanos, and Dedin. The external fertilization and transparency of the zebrafish embryo allows for high-resolution imaging of the germplasm aggregates in the early embryo. So right after fertilization, germplasm is found in single aggregates. A subset of these aggregates are then localized to the furrow and compacted into distal masses, as you can see here with the green. And this is an example of what one of them look like. And then these masses are eventually incorporated into the cell at the 1632 cell stage. As development continues, you can see that the germplasm maintains a discrete boundaries within a cell, and then they are inherited only into one cell. Once the embryo reaches stone, these aggregates burst, releasing the RNAs into a subset of cells. These cells then will be fated to create PGC. So at this early cell stage, when we have these single germplasm molecules, they need to be actively moved to the furrow. And this is done by the use of the cytoskeleton, specifically the microtubules and actin. So we have, the Pellegrini lab has divided this first 20-40 minutes of germplasm aggregation into three different steps. The first step we call pre-aggregation. So this is where in our one-cell embryos, we have these single LNPs that are dispersed across the blastodisc, but they become clustered together in small aggregates while they're being pushed out by these purple astromicrotubules. The second step, which we call furrow recruitment, is where we see these the subset of these RNPs on the furrow. As you can see, they make a nice line, and then eventually they'll compact into the distal end, which we call furrow compaction. And this compaction requires the use of the furrow microtubule array and F-actin. So the Pellegrini lab and other laboratories have identified multiple maternal effect genes that regulate the cytoskeleton to promote germplasm aggregation. One of these genes that regulates this pre-aggregation step is Mochel. So the Mochel mutation maps to the BRCA5B gene. It's a gene that codes for maternally expressed homolog of cervin, a component of the chromosomal passenger complex. So when these mutants fail to divide, on the top, we have a wild type embryo, and you can see the beginning of cellular division. On the bottom, we have a time-matched Motley embryo, and the embryo is not dividing. So the BRC5B is localized both to the furrow. As you can see on the top, we have BRC5B in green, we have microtubules in purple, and it's helping to bundle these microtubules together in the furrow. And then on the bottom, we see that it also localizes to these RNPs. So this is a marker for non-muscle P myosin, and we can use it for to mark germplasm in fish, and we can see it's in red. And then we have BRC5B in green, and they co-localize together. So this is one single RNP. And when they aggregate together in group, they'll actually keep their distinct boundaries. So we're actually able to quantify how large these aggregates are. So when you quantify them in the Motley mutant, on the top we have a wild type, and you can see these larger aggregates. They can go up to 20 RNPs together. And then the bottom we have the Motley, and you can see that the Motley mutant fails to create these higher ordered germplasm aggregates. So one of the questions we wanted to know is BRC5B acting independently, or is it acting within a CPC complex to aggregate germplasm in the cortex? So the CPC complex traditionally regulates key mitotic events, such as correcting chromosomal microtubule attachment errors, the activation of spindle assembly checkpoints, and the construction and regulation of the contractile apparatus that drives cytokinesis. And it's composed of 4 different proteins. So on the top here, we have Aurora B kinase, we have a scaffolding protein intercept, and then on the bottom here, we have this triple helix that promotes the interaction between the cervivin in green and borealin in blue. And all 3 of these proteins are needed to stabilize the helix. So if one is missing, the CPC does not function. So the Motley mutant we have in the lab is actually lacking this interacting region, but it does have this globular head. So it's not able to interact with the CPC. So due to the gene duplication and the maternal function of BRC5B, we wanted to see if any of the other CPC members underwent a gene duplication event and diverged to have a maternal only function. So to do this, we use BLAST and an mRNA baseline expression database for zebrafish, and we investigated the other members of this complex. So on the bottom here, I have the cervinin family, so BRC5A, which is expressed throughout development in this dark green pattern, and BRC5B, which is expressed only maternally in this light green. I also looked at Aurora B kinase and intercept, and they only have 1 copy, and they are both expressed throughout development. Borealin has 2 copies, CDC8 in this dark blue that's expressed throughout development, and CDCA9 in this light blue, which is expressed only maternally. So this expression pattern for CDC8 and CDCA9 is very similar to the expression pattern for BRC5A and BRC5B, and borealin is a direct interactor with cervinin, making it a prime candidate to test if the chromosomal passenger complex is involved in germplasm aggregation. So a little bit background about the borealin paralogs. So CDC8 is composed of 3 distinct domains. On the N-terminal, we have a microtubule binding domain and a coil-coil domain that interacts with cervinin and intercept. Then we have a central region that's on structure that's able to bind to nucleosomes. And then we have a C-terminal region, which contains a dimerization domain that interacts with borealin and sugar-1. So using the domain predictor PFAM, we found that CDCA9 only has this N-terminal CPC interacting domain. I also used RT-PCR to confirm the results from the expression database, and we saw that CDCA8 was expressed throughout development and CDCA9 is expressed maternally. And then I also looked at the expression pattern of CDCA8 and CDCA9 in Xenopus tropicalis, and it also shows the same pattern with CDCA9 expressed maternally. So to look at the role in CDC9 in development and germplasm aggregation, we decided to create a mutant using CRISPR-Cas9. So we targeted the guide RNA to the first active domain, which would be the CPC interaction domain. And we would make a truncated protein like this. We've isolated multiple alleles, but the one I'm going to be using for this talk is UW103. And it's a 13 base pair deletion, and the changes start at the red arrow. And this creates an early premature stop codon, which is less than 52 amino acids long. The nice thing about this allele is that the predicted truncated protein is shorter than the minimum CPC interacting region, which is 109 amino acids long, suggesting this will behave as a null allele. So by mutating CDCA9, we created a new maternal effect mutant. And similar to Motley, CDC9 embryos failed to develop a furrow, So on the top, we have a wild type embryo and 8 cell undergoing cellular division. And on the bottom, we have CDCA9, and it's not undergoing any sort of cellular division. When we stain for cytoskeleton elements such as microtubules and actin, on the top, we have the wild type, and you can see the zone of occlusion where microtubules are being cleaved in the wild type embryos, or you can see this line of actin that forms in these early embryos. And then both the CDCA9 motli and CDCA9 embryo and the motli mutant failed to establish a furrow microtubule array in the zone of occlusion, and they also failed to develop the line of actin in the furrow. So both CDCA9 and motli are essential for the establishment of the furrow in the early embryo. But we also wanted to see if CDCA9 played a role in germplasm aggregation. So we looked at germplasm aggregation in the early embryo, and both CDCA9 embryos and motli mutants failed to create higher order germplasm aggregates. So on the left, we have the wild type, and we can see these higher order aggregates forming. And then in the middle, we have CDCA9, and then we have the motli mutant, and we see a lot of these single aggregates floating around. When we went in and quantified them, we found that around 82% of the time, aggregates were found as RNPs are found as single aggregates in the CDCA9 mutant, and this is compared to 63% of the time in the wild type. So despite having similar phenotypes, CDCA9 and BRC5B are actually localized differently in the cell. So if we're just looking at the furrow, and we know from earlier that BRC5B is in the furrow. So in the picture we have BRC5B, it's bundling these gray microtubules, but CDCA9 is not co-localized with BRC5B, which is surprising for a component of the CPC. And if we just look at intensities across the furrow, you can see that microtubules and CDCA9 are found on the edge of the furrow, and BRC5B is in the center. And then we also looked at the actual microtubules that are aggregating these germplasms, and we noticed that CDCA9 is found on every single microtubule. So if we just look in at this germplasm complex in red and we zoom in on it, we can actually see that CDCA9 is acting as a linker between germplasm and the microtubules. If we just look at fluorescent intensity and we trace it, we can see that it goes to microtubules, then CDCA9, and then germplasm. So what's really interesting about this is if we look at the same stage and we use the BRC5B antibody, we see that the pattern is different. We see a completely different localization pattern. That BRC5B down here is actually co-localized to the germplasm. BRC5B is in green. This is germplasm's in red, and they co-localize 100% of the time. And as the aggregates form, it looks like BRC5B stays with the germplasm and it does not go with CDCA9. So because of this difference, we wanted to see where the other CPC members were in the early embryo. So we looked at intercept, the scaffolding protein, and we see that it co-localizes with CDCA9 on the tips of the microtubules. So if we look at this one aggregate here that I outlined in blue, and we take a Z-stack intensity profile going down through the embryo, so this is the top of the embryo and this is in the embryo, we can see that CDCA9 and intercept are co-localized with the microtubules. And then when we look at this complex in the CPC mutants, we do not see a CDCA9 or intercept complex forming. So on the top, we have CDCA9, we don't see any complex, and then on the bottom, we have the motli mutant, and we don't see any complex. So this is not really surprising because, as I mentioned earlier, when one of the proteins is missing from that triple helix, that CPC complex can't be stabilized. And then we also looked at Aurora B kinase, and we saw the same pattern where it localizes with CDCA9. And this is just another intensity tracing of an aggregate with the top of the embryo here. And then going down to the blastodisc, you see they co-localize together. And similar to the intercept data, the CPC mutants do not have any CDC9 or RB kinase complexes. So we know now that a CPC complex is found on the tips of the microtubules to promote germplasm aggregation. We wanted to know if the CPC complex was necessary for the interaction of BRC5B with germplasm in the early embryo. So what we decided to do was stain the wild type and CPC mutants for BUC, which is a component of germplasm here in red, BRC5B, which is cervivin in green, and look at colocalization. So in the wild type embryo, you can see they colocalize 100%, and you can also see these larger aggregates beginning to form. And then in the CDCA9 mutant and the Mochel mutant, we still see them colocalizing 100% of the time, and we see smaller aggregates. So it allows us to start building a model where we think we have a CPC complex, but the germplasm is only interacting with the BRC5B globular head. And when CDCA9 is missing down here, as you see, we miss the light blue protein, the CPC complex can't form, so we don't get, but we can still have germplasm interacting with BRC5B. And in the Mochel mutant, where we're lacking that tail that allows for the interaction, but we still have the globular head, we're still able to get BRC5B and germplasm interaction. And we wanted to know how early this interaction occurs. So these embryos at 40 minutes, the astromicrotubules are growing out. Maybe the astromicrotubules play a role in this action. So we decided to look at 25 minutes. And you can see here on the gray, we have the beginning of the formation of the spindle. It's about to start outgrowing. And if we look at the edge of the cortex, we can actually see BRC5B and BUC already interacting. So this tells us that the CPC is not necessary for the interaction of BRC5B and germplasm, and this interaction occurs early in the embryo, but it is necessary for the interaction with microtubules. So what I have here are 3 3D reconstructions of germplasm molecules in wild type, CDCA9, and Mochel. And again, we're staying for BUC in green, BRC5B in red, and the microtubules on purple. And I'm going to play this 3D reconstruction, and it's going to rotate around. And what you guys will notice is that the germplasm complex is localized to the microtubule. So that as you see it rotating, you can see it's nicely localized to this microtubule. And then that kind of lets us kind of start building a model like this, where we have a CPC complex and it's interacting with microtubules, either by maybe a microtubule binding domain or by a kinesin. And then when we look at the CDCA9 mutants or the BRC5B mutants, what you're going to notice is they're not localized to microtubule. It's almost like they're sitting in the middle of a mesh of microtubules as these microtubules are going by. So I'll play CDCA9 first. So as you can see, it's just sitting in the microtubule, it's not being localized in the CDCA9, and then in the Mochel mutant, you see the same pattern. I'll let it rotate one more time. So that allows us to kind of infer this. So in the CDCA9, when we're missing the blue CDCA9 protein, the CPC complex can't form and can't attach to the microtubule. In the Mochel mutant, where we have the globular head of BRC5B, but we lack the interacting domain, it cannot interact with this intercept or CDCA9, cannot form a CPC, and it cannot interact with the microtubule. So because this interaction of BRC5B and germplasm occurs really early in the embryo, we wanted to know, does it continue into the other stages of germplasm compaction, such as furrow recruitment and furrow compaction? So as I mentioned earlier, we know that BRC5B has a role in the furrow with helping to bundle these microtubules. So the microtubules are in purple, the BRC5B is in green. And we also know that the actin has this dynamic wave action that helps compact these germplasm. So here is a stain for a BUC, a component of germplasm in green, actin in blue. And when you do a 3D reconstruction and flip it on the side, you can see this dynamic wave action. And you can see BUC hanging down from the actin. So it's possible that there needs to be a linker between germplasm and actin. And in the cortex, BRC5B is known to be a linker between actin, microtubules, and germplasm. So it's possible that it could be a linker between actin and germplasm in the furrow. So we did a triple immunostain, and we did a 3D reconstruction of the furrow and flipped it on the side. And as you can see, we see this dynamic blue actin wave, and we see germplasm hanging down from it. When you take away the germplasm channel and just look at BRC5B, you can actually see BRC5B hanging down from the actin too. And then if we go in and look at it and do a intensity readout going from the top of the cortex downwards, so the top's here, this is in the cell, we can see that it goes actin, BRC5B, and then germplasm. So this suggests that this could be acting as a linker between actin and germplasm. We also wanted to know if it stayed with the germplasm once it's been compacted into the distal mass. So this is a 4-cell embryo, and you can see 1, 2, 3, 4 clusters. When we zoom in on them, we can see 100% colocalization of germplasm and BRC5B in these clusters. We are currently investigating how long BRC5B is located with the germplasm. So in general, this data has allowed us to put together this model that has some questions, but also is really informative. So we have these astromicrotubules that have a CPC complex on it. And as it grows outward, it runs into these germplasm BRC5B complex. A swap occurs that lets this BRC5B interacts with the CPC on the astromicrotubules, and that pushes it outwards. Then once it gets to the furrow, the germplasm and BRC5B complex disassembles. And then it's also, the BRC5B is also able to compact into these germplasm masses. So in summary, the assembly of germplasm complex with BRC5B is not dependent on the CPC, and the attachment of germplasm complex to the microtubules is dependent on the CPC. So this would be a novel role for the CPC. So I would like to thank my mentor, Dr. Pellegrini, the laboratory, Christina, who does a lot of the amazing imaging of germplasm RNPs, Brady, who helps me with the intensity profiling. All my imaging is done at the Newcomb Imaging Center. And then we also like to thank the Mullins Lab, providing us with the Buck antibody. And now I will take questions.
Thank you, Cara, for the great talk. You're on the time. So here I'd like to mention that if anyone has a question, so you can enter a question into the QA box, so that way I can just read out. So I think I can start with the first question.
Awesome.
The CDCA8 can rescue the CDCA9 phenotype?
So we have not had success in that experiment yet, because the role of, because you can't just inject CDCA into the embryo, you have to culture them first. So that's something we've been working on.
Okay, I see. So how much similar between the CDCA8 to CDCA9? So
they're not actually that similar. The start of them domains are similar, but when you get to the C-terminal, they're not. So I ran a conserve calculation, which will look at how conserved amino acids are through evolution. And when you run it on zebrafish CDCA9, it would only pick up the most conserved region, being the N-terminal, where it interacts with the CPC. But when you look at it, CDC8, you get both of them. And then when you look specifically at the C-terminals across fish species and other creatures that have it, such as xenopus, chicken, you really don't have any high homology regions in that C-terminal region.
Okay, great. So there's a question from the chat from Jessica. So ask, any idea how the CPC is getting to the ends of the microtubule? And from your imaging, it seems to bind the sites. Is there a kinesis involved? Kinesin?
So there is a kinesin that's involved in getting germplasm into the furrow. It's from the Marlowe lab, and I believe it's a kinesin 4, but we don't know if it has a role in the pre-aggregation step. But I also have another maternally duplicated, I believe it's KIF23, it's ZGC56321, and I made that maternal mutant, and it has the exact same phenotype as Motley and CDCA9, where the embryos failed to divide. So that could also have a role in germplasm aggregation. So there are a couple possibilities for what kinesins could be doing this.
Okay. Oh, follow Jessica's question. Does the CPC have any effect on the dynamics of the microtubule?
So again, that's something that we would like to do. My next step in the project, which I'm trying to do right now, is to start doing live imaging with the CT and a EMTB transgenic microtubule line. So I have some preliminary results from it, but nothing that I feel comfortable sharing yet.
Okay. The other question from Nico asked, a great talk. It seems like in your mutant cells, there were more microtubes accumulating around the germplasm compared to wild type. Is that true?
I have not looked at that yet, so I cannot speak to that.
Okay. David asked, much of the germplasm remains in non-higher order structure. What is the function of the higher structure?
So this is something that we have been really interested in the Pellegrini lab. So the germplasm itself is intrinsically attracted to itself. So even in the Motley mutants and the CDCA9 mutants, we actually see a few higher order aggregates form. But we think the role of the higher order aggregates is that they actually get moved to the furrow. So we know that we have about 1,000 RNP molecules in the first furrow on both sides, so around 2,000 total. And then the second furrow, we have about 500. So that's a lot of RNPs that need to be actively moved to the furrow. So we think these larger aggregates allow it to happen. It's a very dynamic process.
Yeah, okay. Okay, I think we have gone through all of the questions, and thank you again for a great talk. Maybe we can go to next speaker.
Awesome, thank you for listening.
Yeah.
Okay, wonderful. So I'm going to present the next speaker. It's Mr. Brandon Carpenter. So Brandon received his Bachelor's of Science in Biology with a major of chemistry. Then he went on to complete a master's degree in cell and molecular biology. Eventually, that led him to be interested in developmental biology, where he received his PhD in the Department of Cell and Developmental Biology, studying kinase motor and how they regulate trafficking and function of effector proteins of the Hedgehog pathway, in particular GLI1 transcription factors. He's also won a numerous of awards that are awarded for him to complete his current work that is being done at Emory University under David Katz. He's received a fellowship in research and science. He's also been awarded the NRSA F32 NIH award. And this award was given to him so he can look at the epigenetic reprogramming and how between different generations in the context of development and how this affects fate decision making. And with that, I'll turn it over to Brandon.
All right, can everybody see my screen and hear me loud and clear?
Yes.
All right, perfect. So thank you, Soledad, for that introduction, and thank SDB for giving me the opportunity to tell you about my research. And the story today is going to be about this early stage C. elegans larvae that you see here, which is largely composed of somatic tissues. But these foci you see are a germline gene that's being misexpressed throughout these somatic tissues. So how does this happen? Well, what happens is when you inappropriately inherit chromatin state, you get disruption in germline versus soma. So what we're interested in the Katz lab is trying to understand how chromatin state is inherited from one generation to the next, and what this means for development, what it means for C. elegans development, vertebrate development, and also human disease. So let's warm up with a little orientation to chromatin. It's DNA that consists of these histone proteins. DNA is wrapped around these histone core proteins. Modifications or marks, I'm going to use those words interchangeably, to the N-terminal tails of these histone core proteins can regulate accessibility to the DNA itself. So some of these marks we consider active marks. They open up chromatin and they're generally associated with active transcription. There's other marks that are repressive marks. They close chromatin down and make it inaccessible. And these are generally associated with repressed transcription. So how are these histone modifications typically laid down? Well, they're typically laid down in a transcriptionally dependent manner. So here we're looking at an example of H3K4, which is an active mark being laid down. It's coupled to transcription. You have pol2 chugging along. This is the compass complex with set2 being the H3K4 methyltransferase. So as the gene's being transcribed, set2 is adding K4 at these histone tails. This is creating an open active state that can then be inherited not only through mitotic divisions, but from one generation to the next. And this is important for development. So as you can pretend like you're in a muscle cell, this is a muscle gene, you're transcribing, you do go through a mitotic division. You don't have to reopen up chromatin to create this muscle specific cell fate. But you can imagine what happens when you have to change cell fates. It can present a problem, right? And probably the most extreme example of this is fertilization. When you have to go from a highly specialized oocyte and sperm that's expressing lots of germline genes that have acquired these active chromatin marks, they have to fuse together and make a totipotent zygote that can then become any cell in the embryo. So how do you get around this problem? And this is something that we've been interested in the CATS lab for more than a decade now. And you have to reprogram that epigenetic landscape and reset it. And this is done through the synergistic activity of two histone modifying enzymes, SPR5 and MET2. So SPR5, both of these enzymes are deposited in the oocyte where they go to work. It's the way I like to think about it. And SPR5 is a H3K4 demethylase. So it's removing H3K4 methylation. H3K4 methylation, if you can remember, is an active mark. So SPR5 removes that active mark. MET2 is an H3K9 methyltransferase. And what it does is it adds H3K9 methylation. And H3K9 methylation is a repressive mark. So I think about these two enzymes like a switch, where SPR5 sort of comes in, it removes the active mark, MET2 adds a repressive mark, and this resets the epigenetic landscape. And it prevents this problem of having this open chromatin coming through into the zygote. So C. elegans, while it's got to fix that problem of resetting the epigenetic landscape, it also has another interesting problem to get over. And that is, in the presence of this reprogramming event, how do you specify germ cell fate? And the germ cells will be in blue, outlined these blue dots, because these germ cells we know are transcriptionally inactive. So how, in the absence of transcription, where you're not acquiring new marks, and in the presence of this reprogramming event, do you establish germ cell fate? And the answer to this came from two beautiful papers from Bill Kelly and Susan Strom's lab in 2010. And it's through the maternal deposition of this enzyme MES4. MES4 is an H3K36 methyltransferase. It's deposited into the oocyte, just like SPR5 and MET2. But what it's doing is this really unique job. It recognizes H3K36, an active mark that had been laid down at a specific set of germline genes that was transcribed in the parents. And it is recruited to these germline genes where it maintains K36. It maintains this epigenetic memory of that germline gene expression. And this helps establish germ cell fate. And if you lose MES4, you get a happy, healthy somatic worm, but you absolutely have no germline. They're completely sterile. So the story I'm going to tell you about today is how you have to balance these histone modifications, the activity of these histone modifying enzymes, to both erase the slate and also maintain some memory. And it's this balance that helps you specify properly somatic cell fates versus germline cell fates. And the story is going to be about what happens when this breaks down. And we're going to look at this in the context of SPR5 MET2 mutants, where we lose that maternal reprogramming. We can't reset the epigenetic landscape. And I'm going to go ahead and give you the spoiler alert. This beautiful role that MES4 is doing in creating this transcriptional memory of germline genes from one generation to the next, and the germline becomes a problem. Now MES4 starts maintaining active chromatin marks at those same germline genes in somatic tissues. And what this leads to is ectopic expression of those germline genes in a range of debilitating developmental phenotypes, including a severe developmental delay. So when I stepped foot in the lab as a postdoc and a classical developmental biologist, we knew we had just published these double mutants, and we know that when you knock down SPR5 and MET2, you get this synergistic increase in accumulation H3K4 methylation across a generation. But what we didn't know is to what extent this affects somatic development. We also knew that there were hints that germline genes may be misexpressed in somatic tissues, but we didn't know to what extent. So I started off by just doing like every classical developmental biologist does, is just staring at your organism for a long period of time to see if they're screwed up. And the way I did this is a DIC imaging synchronized lay, where I took hermaphrodites of wild type, which is N2. Whenever you see N2, that's the wild type strain, and C. elegans, single mutants, and I let them lay for about 2 hours, and I took the hermaphrodites off. So I had this nice synchronized population of progeny, and I monitored their development and did snapshots every 12 hours. And what I saw when we did this with single mutants, by 72 hours, the progeny from that synchronized lay are happy, healthy adults laying their own progeny. And we've quantified this down here in this bar graph to show percent adult progeny. But what we noticed, which was really interesting, is none of the SPR5 MET2 progeny made it. In fact, many of these are kind of in this early L2 larval stage, but it's not a complete arrest, because if you let this plate sit out and you look 7 days later, many of these progeny have made it to advanced larval stages, and about 4% of them can make it to adults. So I want to drive home the fact here that this is truly maternal effect sterile phenotype, because first generation mutants are fine because their parents or heterozygous for SPR5 and MET2, they could deposit those enzymes into the oocyte and reprogramming happened. But these first generation mutants, which are anatomically normal when we look at them, they can't provide those enzymes for their progeny. So all the data that you're going to see today are with these SPR5 MET2 progeny that lack this reprogramming. So we see this debilitating developmental delay. We were interested, I told you we had hints that germline genes may be expressed in somatic tissues. We wanted to do the RNA-seq to really go after and look, and we wanted to do this in the somatic tissue. And to do this, we took advantage of the L1 larvae stage, which in this stage is almost entirely composed of somatic tissue. There's only 2 germ cells. And to bulk up our material, we let these L1 larvae hatch without food. So when we do this, that also stops germline gene transcription. So we're in a largely somatic tissue, should not be expressing any germline genes. And when we did the RNA-seq, we were blown away. Not only do we see enrichment for germline genes, when we looked at those germline genes, they tended to be germline genes that were bound by MES4 that are bookmarked from one generation to the next. And here you're looking at a heat map of the RNA-seq experiment, and the colors represent log2 fold values, blue being upregulated compared to wild type, yellow being downregulated compared to wild type, with single mutants and double mutants. And what I hope you can appreciate is while we start to see some blue, we start to see some ectopic expression of these germline genes in this somatic tissue, we see this beautiful synergistic increase in this ectopic expression in SPR5 MET2 double mutants. And what this suggested to us is that this MES4 bookmarking program is coming on in the somatic tissues of these mutants that are deficient in this maternal reprogramming. So the next step, we wanted to visualize this. So we turned to single molecule FISH, where we can detect endogenous transcripts. And here we chose to look at 2 to start with, HTP1 and CPB1. These genes were upregulated MES4 germline genes in our RNA-seq data set, and they're well characterized in the literature. So we asked, where do we see this mRNA being expressed? And the top panels here are going to be in a wild type L1 larvae. And what I hope you can appreciate is the 2 cells that you see these mRNAs being expressed are Z2 and Z3. We see this in both cases with HTP1 and with CPB1. Now, when we look in our double mutants, we see a completely different story. Now we see ectopic foci throughout the somatic tissue. And this was the star of the show. This is who you saw on the first slide. We also see this with CPB1, albeit to lower levels. So not only do we detect this in the RNA-seq, we can visualize that these MES4 germline genes are coming on throughout the somatic tissues. So the next step, we reason that if MES4 is causing the problem, if we could knock down MES4 maternally, could we rescue the transcription, the ectopic transcription of these MES4 germline genes? So to do this, we turn to just a real-time PCR experiment in RNAi, where we can feed C. elegans, a bacteria, with RNAi construct that can knock down a gene of choice. In this case, we're going to knock down MES4 maternally. And I chose a handful of MES4 germline genes that you see in blue that were ectopic in our RNA-seq data set. I also chose a handful of control genes, which included housekeeping, somatic-specific genes. And what I hope you can appreciate as we go from wild type to double mutants, all of our MES4 germline genes are ectopic. So this fits very nicely with our RNA-seq data. But when we look at double mutants, when we look at the control genes, there's no change in those genes. When we look at SPR5-MET2 hermaphrodites that we put on MES4 RNAi, and we look at the expression of these genes in their progeny, what was striking is that all of these genes come down, and all but one come back down to wild type levels. So by knocking down MES4 maternally, we rescued this ectopic expression of MES4 germline genes. So we were interested if it's the transcription that's causing the problem, what if we knock down MES4 and look at the developmental delay again? Do we see a rescue? And to do this, I'm combining that developmental time course that I already showed you with RNAi. And I've already showed you, and here we're looking at 72 hours after that synchronized lay. This time we did it on RNAi. So we have control RNAi where we've synchronized laid wild type embryos. And by 72 hours, 100% of those progeny have made it to adults. So instead of the bar graph, now you're going to see that percentage in the top part of the box here. As I showed previously, SPR5-MET2 on control RNAi, almost none of these make it to adults by 72 hours. But when we put these worms on MES4 RNAi, what we see is pretty striking. We know the MES4 RNAi is working because I told you that if you maternally knock down MES4, the progeny are completely sterile. And we see that with these wild type worms that were placed on MES4 RNAi. And then what we see with the double mutants is striking. Now more than 90% of these worms make it to adults just fine. In fact, there's very little difference between these two worms when we looked at the plates. It was quite striking rescue. So this allowed us to kind of think about the model, and it's, in the absence of SPR5 and MET2, you let this ectopic K4, you don't remove the K4 and add the K9. This ectopic K4 allows MES4 that encroach on those MES4 germline genes in somatic tissues, maintain K36, and ectopically express MES4 germline genes. And if this is the case, we would expect that if we went into these MES4 germline genes in the somatic tissues, we should see evidence of MES4 activity, which is maintaining H3K36. So we looked at H3K36 chip in wild type worms and double mutants. The orange bars here are transcriptional start and insight. These are the reads from the ChIP-seq experiment, normalized reads per kilobase million. And you're looking at MES4 germline genes from top to bottom here. And what I hope you can appreciate is there's more blue. We have higher levels of H3K36 in these double mutants. So the prediction one of our model is met. We do see this ectopic increase in H3K36. This is perhaps one of my favorite experiments. You would predict that the developmental delay in SPR5-MET2 progeny is dependent on H3K4 methylation. So I was teaching an epigenetics course at Oglethorpe University, and a student raised their hand while we were hashing at this model and said, Dr. C, doesn't your model where K4 doesn't get erased, isn't K4 the problem? If you could knock down K4, could you also rescue the phenotype like you do with MES4? And he was on to something with that experiment. We were thinking about it, but for the student to come up with that independently, it was really cool. So we came up with a way to do that. What he was getting at is if we could lower the levels of H3K4 methylation, could we also rescue this developmental delay? So to do this, we knocked down set 2, and I told you about the compass complex. It's responsible for adding H3K4 methylation. So Jovan, the student that came with the idea, I made him do the experiment. And he was happy to do this. And after the fact, he was really glad because we completely rescue the phenotype, the developmental delay, similar to what we see with MES4. So this suggested that, this H3K4 methylation is required for this developmental delay phenotype. We also expected that if you went into a wild type L1 larvae and you looked in the somatic tissue at these germline genes, we should see evidence of this reprogramming where SPR5 is removed and you add K9. And lucky for us, Lisa Petrella had just published this beautiful paper where she did H3K9 dimethyl chip on wild type L1 larvae. So we reanalyzed that data in the context of these MES4 germline genes. Here you're looking at IGV shots of individual loci, with the arrows being the promoters and then control genes. We also looked at all the MES4 germline genes. What I hope you can appreciate is we see evidence of H3K9 dimethyl at the promoters of these germline genes. We don't see this in control genes. So it suggests that these things are being reprogrammed and helps keep them off in the somatic tissues. So I want to step back and talk about in the last couple of slides how we're thinking about this model and what it means for vertebrate development and human disease. First, let's start with C. elegans. So as you're making a germline and you're transcribing germline genes, they acquire transcriptional baggage in the form of these active marks. You've got to deal with that as you come through the gametes to fertilization. And it's through this balance of activity by these histone modifying enzymes where you both remove active marks and add repressive marks, like in the case of SPR5 and MET2. And it's absolutely critical for resetting the epigenetic landscape. But at the same time, you have to maintain transcriptional memory of certain genes that help specify germ cell fate. And this is done through the activity of MES4. In the absence of SPR5 and MET2, now SPR5 cannot remove the K4, MET2 can't add K9. And what happens now is that beautiful role for MES4 spills over in the somatic tissues, and you ectopically maintain K36 at these germline genes. And this leads to severe developmental delay. Now, we know that there's these beautiful mechanisms. Once the zygote genome turns on and it starts expressing its own genome, there's somatic mechanisms that help reinforce this. And this is beautiful work from Craig Mello, Lisa Petrella, and others. But in the absence of SPR5 and MET2, whether these somatic mechanisms are there or not, it doesn't matter. You still get this ectopic K36 leading this severe developmental delay. So what we've shown here is that if you don't properly filter the inherited chromatin state, you can get transcriptional programs expressed in the wrong cell types. So we think that how you're getting a developmental delay is the somatic program just can't properly read out with this underlying interference of germline transcription. And the reason why we think this is important is this mechanism seems to be conserved in vertebrates and humans. We've started to do these experiments in mice, and what we've shown, SPR5 homolog in humans is LSD1. MET2 homolog is SETDB1. Maternal loss of either of those results in the embryonic arrest of the progeny at the 2-cell stage. So we took this a step further and we performed RNA-seq on those arrested 2-cell mouse embryos that lacked that maternal reprogramming. And what we saw was they failed to turn off 1,800 of the 3,000 maternal germline genes. We also did a neat genetic trick where you can basically, partially reduce the activity of LSD1 in this reprogramming. And what happened was we bypassed that 2-cell arrest. These progeny made it to adults, but they display neurological defects in learning and memory. And what's really exciting about that is the phenotypes that we're seeing in the mice and even some of the phenotypes in the worm overlap with the human patient phenotypes that have mutations in these same enzymes. So we think when you look at these patients, they have ranges, and these are patients. Yep, last slide. So when you look at these patients that have mutations in LSD1, they have global somatic defects, a lot of craniofacial defects, developmental delay. We think, based on what I've shown you in C. elegans, is that one way you can get these global effects is if you have the wrong transcriptional program at low levels interfering with the readout of the somatic program. So we hope that the mechanisms that we're onto in C. elegans are relevant for the human diseases. So with that, I'll acknowledge the CATS lab. David Katz, a tremendous mentor. We have a terrific lab. If you guys don't know the CATS lab, we love teaching and we love bringing our research to Oglethorpe University, where we get to bring exciting experiments to those students. And as a result, we have this lab full of talented undergraduates that have come from Oglethorpe and Emory that really gravitate towards the worm research and help crank out the science that you saw today. So with that, I'll take any questions, and thank you for your attention.
Okay, thank you, Brandon, for the very beautiful talk. Wow, there's a couple of questions now. There's one from Jessica Fieldman, and she wants to know have you looked at the somatic tissue in your double mutant? Is it normal beyond just being delayed? Are fates specified correctly in is differentiation normal? And actually, this was my question precisely as well too. Yeah. So I'm very glad someone else asked it.
You and Jessica talked before. No, that's a great question. I'm on the market right now, and one of the things that I'm super excited about is going into these somatic tissues and looking at that. I think that we know that there's more defects. So for instance, I didn't talk a lot about the vulval system. These things have protruding vulva. There's distal tip cell migrations. There is a smorgasbord of somatic defects, Jessica and Soledad. So I am super interested in going in and trying to understand what's going on in those somatic tissues. How do you go from an inherited chromatin state now to these developmental phenotypes, especially now that we know that there's patients walking around with mutations? We think it's directly relevant, but we are just now starting to get into those somatic tissues and look specifically. So stay tuned.
Yeah, I would be really interested to see if there's other MES. I mean, my assumption would be that there is other factors like MES4 and you know, that help in this reprogramming, and how could we exploit that in the laboratory or for therapeutic purposes?
Absolutely, cool. Yes.
We're still waiting. I wrote in a lot of notes because I actually learned so much from your presentation. I am not very familiar with the epigenetic field, but I became quite interested in it because I study some senescent cells, and it would be, it's of interest to me to understand how the epigenetic landscape changes with time in these cells, and how that correlates with their behavior.
Yeah.
So
one thing that we're interested in, and this is something that Dave's really interested in, is the behavior, right? Can we start doing some of these neurological behavior assays in these worms? So these are directions that we're really interested in heading with the lab.
Okay, great. So now we have a question from Ann Corsi. She says, absolutely beautiful talk. Do any of the de novo human mutations occur in amino acids that are conserved in the worm proteins that you can model in C. elegans?
Yeah,
that's
absolutely. Yeah, so that's a great question. One of the things that I'm really interested in is, you struck a nerve here, because one of my future directions is to start to do that. And lucky for us, C. elegans just got recognition in the undiagnosed disease network. It's a model system now to study these mutations in human patients and how they're functioning in C. elegans. So we know where those mutations are. One of them is conserved. What I'm doing right now is trying to humanize the C. elegans and take out SPR5 and put in the human LSD1 and then make the exact mutations. And then ask, I have this beautiful system now where we can look at MES4 inheritance system to see how these things are functioning. So those are directions that are on the list of priorities to do. And I think that as we start seeing more and more of these patients that have mutations in these histone modifying enzymes, we need to get them into the worm. And if they're not conserved in the worm, we can humanize the worm. And in vivo biosystems is getting really good at doing this and relatively quickly. So those technologies will be here soon.
There's a second part to Anne's questions. Also, are there any sex-specific differences in worms? And if you look in, I think it's in the mutants, do you see anything different with your SPR5 MET2 mutants in the males?
That's a fantastic question, and we have not looked at all. We have been 100% in the hermaphrodite. It's a great question. We just don't know. My guess is maybe, I'm sure, if we look closely, but we need to get there first. We haven't done that. That's a great question.
Okay, well, we don't have any more questions coming in. So I would like to thank the speakers and all the audience for coming to the SDB Postdoc Seminar Series. Oh, wait, actually, we have one question that rolled in, and since we have time, I think that I'll go ahead and read it. I think it takes time for people to type. So Nicole Edwards would like to know if, do the human patients have reduced fertility or sterility with the mutations?
I saw that question come up, and I know the somatic phenotypes. I was just looking at them today, and I don't know about sterility. That's a great question. I can find out in 5 seconds, but I don't know right now. That's a great question. I don't know the answer to it.
Okay, well, then I think that is it for now. Again, thanks for both of our speakers, Cara and Brandon, which did a wonderful job. And thank you all for tuning in, and I hope everyone has a wonderful weekend.